program for partially funding this project, as well as the OSU College of Forestry for allowing me to do my study in the Vera McDonald Dunn Research Forest. Finally, I would like to thank August Jackson, Lincoln Best, and Anthony Malthopoulos for assistance with identifying bees as well as other pollinators. So oak savanna is a unique ecosystem to the inland west coast of North America. It can be as far south as Southern California or as far north as British Columbia, Canada. But what all these locations of oak savanna have in common is that they have widely spaced trees and ground cover made up of grasses and herbaceous plants, as well as the occasional shrub. Oak savanna is often a transition zone between oak woodland and wet prairie, but it has characteristics of both. So unlike an oak woodland, the trees are much more widely spaced, and this allows sunlight to reach the ground, and that's why all these grasses thrive that wouldn't ordinarily in a more shaded, dense woodland. And unlike a wet prairie, oak savanna notably has oak trees. That's why it's called oak savanna. And it also tends to be a bit drier. And the trees are primarily Oregon white oak, but there can also be occasional big leaf maple or Douglas fir or ponderosa pine as well. So in the understory, the grasses historically were native bunch grasses, such as this roamer's fescue, but now a lot of oak savanna fragments, unfortunately, have been taken over by invasive false brome, like this site in that photo. There's also a lot of native herbaceous plants, such as this western buttercup. They grow in patches in between clumps of grass, and it creates this very diverse mosaic of different species below the oak trees. There's also some shrubs like snowberry or poison oak in this photo that can actually be a shrub or a vine, but it usually grows as a shrub in oak savanna. And in addition to this diverse plant community, oak savanna also supports 14 species of native reptiles, 31 species of native mammals, and 48 native bird species in Oregon alone, as well as numerous invertebrates, including pollinators. So oak savanna is an important habitat for some species that might have a limited range or are threatened in other ways. So up top here is the Camas pocket gopher, and below is the gray-tailed bull. These are both rodent species that are endemic to the Willamette Valley, so they don't live anywhere else, just the Willamette Valley, and they spend their entire lives in the oak savanna. The Camas pocket gopher feeds on the bulbs of Camas, which is a native flower. Then we also have the Oregon vesper sparrow above and below the street-toed lark. The vesper sparrow is a U.S. Fish and Wildlife Service species of concern, and the street-toed lark is a candidate for listing under the Federal Endangered Species Act. Then we also have two butterfly species. The Fetter's blue butterfly is listed under the Federal Endangered Species Act as endangered, and the Taylor's checker spot butterfly is a candidate for federal listing. And oak savanna is important habitat for all of these species. So Oregon white oak, the main tree species in oak savanna, is a slow-growing tree. It likes sunny, dry areas, but it also occurs in the same areas as Douglas fir, which is a much faster-growing tree, and it can crowd out the oak trees if given the right conditions, and can even block out sunlight. So historically, indigenous peoples in the Willamette Valley would burn the oak savanna understory in order to maintain habitat for important plants that acted as food and provided other resources for them. And that would also have the added benefit of clearing out all these Douglas fir seedlings. So pretty quickly after fire, the native herbs and grasses will grow back, and that keeps the understory clear of Douglas fir trees that would otherwise crowd out the oak trees. But after European settlement, land management has focused a lot more on excluding fire altogether, and indigenous peoples have not been allowed to conduct those routine burns in oak savannas. So when fire is excluded, you get something like this, where the Douglas fir trees grow up fast. It creates a denser forest with a shaded understory, and different understory plants can grow there. None of the grasses usually, and the oaks will eventually start to decline because they can't get enough sunlight. So largely because of those fire suppression practices, 90% of the oak savanna and other oak-related habitats in the Willamette Valley are degraded or destroyed. So this map shows the Willamette Valley and the oak-associated habitat in 1850. You can see all that green area is oak savanna or oak woodland. And then in 2018, that area has significantly decreased, and also the remaining habitat is in very small fragments. Now, urbanization and conversion of land to agriculture has also contributed to this decline, as well as degradation of oak savanna can occur when invasive plants take over, such as invasive blackberries or English ivy, to name a few. But the good news is there's a lot of interest in restoring oak savanna. And an important part of conservation is the flowering plants that make up part of the oak savanna understory. So if you're a restoration group and you want to choose what flowering plants to plant when you're restoring an oak savanna, you want plants that are attractive to pollinators because, one, it'll support the local pollinator community, which is also a goal of many conservation projects. And also that means that those plants will be able to reproduce because pollinators will visit them to allow them to produce seeds. So this benefits both pollinators and plants. So past studies have shown a number of native flowers that historically are associated with oak savanna. These are the camas, short for sea blush, Canada goldenrod, Douglas aster, farewell to spring, western buttercup, trailing blackberry, globelia, and bare stem biscuit root. So all of these plants have been shown in past studies to be very attractive to pollinators. And they also happen to grow in oak savanna. So my objective in this study was to determine which oak savanna flowering plants are good food sources for native pollinators. And my hypothesis was that native flowers, like the ones I just showed on the previous slide, would be preferred by the pollinators in oak savanna. I conducted my research at the OSU's McDonald Research Forest, which is just north of Corvallis, Oregon. Corvallis there is in yellow on the map. And the sites that I conducted my study at are all within this black rectangle. So now that you can see the extent of the forest and where it's located in regards to Corvallis, I'm going to zoom in on that location. So this is a close-up of that black rectangle, and I did put my research sites in 2 oak savanna fragments. So the first fragment was about 600,000 square feet, and that's outlined in yellow there. And I set up my research sites 1, 2, and 3. And then in the second fragment, which was about 70,000 square feet, I set up my 4th research site. And that was just because there wasn't enough space in the first fragment for 4. So at each of these research sites, I set up 5 transects. Those are shown in green. They were each 15 feet long, 15 feet apart, and 3 feet wide. And then I also decided that I was going to collect pollinators within a 75-foot radius of the center of the site, which is marked here in the X. So brown area is where I collected pollinators, green is where I looked at plants, and X is the center of the site. So I did my research sampling from March 8th to September 14th of 2025. So for recording flowering plants, what I did at each site, I walked along these 5 transects and recorded how many blooms of each flowering plant species there were, and then I recorded what species was there. Then I captured pollinators using a hand net. That's me there catching a bee or some other pollinator probably. And I would walk around that whole 75-foot radius for 20 minutes in the morning and 20 minutes in the afternoon at each site. And then those insects I caught, I pinned them. I identified them to the order level, so whether those are a bee or wasp or a fly or a beetle or something else. And then I identified the flies, beetles, and bees and wasps to the family level, and then I identified the bees to the genus level. And that's where I used the dissecting microscope to look really closely and see these tiny details. A lot of it has to do with the veins in their wings. It's very cool. So for my data analysis, first I calculated the total number of I used the data that I had about insects and what plants they visited to create a pollination network, which I will show you later. Then I calculated the total number of blooms that I collected of each flowering plant species. So that was just how many flowers were in bloom over the entire time at all the sites. Then I did that same thing for all the insects that I collected across all of my insect surveys. So that's just the total number of insects I collected visiting each plant species. And those 2 pieces of information, I was able to calculate the insect visitation rate for each plant species. So that was just the total number of pollinators that visited a plant divided by the total number of flowers of that plant. And that's pretty much just a measurement of how much insects liked that plant. So what I found across that entire summer, all of my 4 sites, I found 37 flowering plant species from 32 different genera, and 20 of those plants were native to Oregon. 17 were introduced from somewhere else. And the most abundant of those were common St. John's wort that had 1,330 blooms. Then we have our Canada thistle with 587 blooms, our cleavers with 486 blooms, hedge parsley had 478 blooms, large leaf sandwort had 370 blooms, oxeye daisy had 250 blooms, small flower nemophila, this little fuzzy flower there, had 227 blooms, and European blackberry, that's an invasive blackberry species, that had also 227 blooms. And our native blackberry species, trailing blackberry, had 196 blooms. So I also collected 313 different insects that were visiting flowers from 5 orders, but the main 3 orders that I collected were Hymenoptera, that's bees, wasps, ants, and sawflies. Sawflies, if you don't know, they're these guys. They're cool, but I didn't catch any of those actually on plants, but I did find some that were on the ground. Then I also did not collect ants because they're very tiny and difficult to pin, so I made that decision. I also didn't collect any pollinators from the order Lepidoptera, which is butterflies and moths, because they're very difficult to keep the scales from the wings from rubbing off before identifying them. So I collected 182 insects in total from the order Hymenoptera, bees and wasps, 76 from order Coleoptera, which is beetles, and then 43 from order Diptera, which is flies. So looking into that order Hymenoptera, of those 176 bees or insects, 97% of those were bees. So the vast majority, I found, I think about 9 wasps. 3% of that were wasps, but very much mostly bees. And those came from 5 different families of bees. So Anginaceae, there are 5 bees, 81 from family Apidae, that includes bumblebees like that one. Calididae had 11, Halictidae had 57, and Megachilidae had 22. Now, I mentioned that I collected some insects that were on the ground, or some of them were also in flight or on the leaves or plants of stems, that they were not foraging on flowers. And this graph shows the bees that I collected from 14 different genera. So there were 14 different bee genera that I collected at these sites, and most of them were visiting flowers. Those are the blue portion of the bar. On the y-axis, that shows the number of bees that I collected, and the blue portion shows the number of bees I collected on flowers. Then the orange portion of the bar shows the number of bees that I collected that were not foraging on flowers. They were flying around or sitting on the ground or sitting on a different part of a plant. And you can see there's a lot of blue in this graph because most of the bees were visiting flowers. But there are 2 genera here where the majority are not visiting flowers. That's the genus Andrena, about half and half visiting flowers and not visiting flowers. And then the genus Nomada had most of the bees actually were not visiting flowers. And I'll talk a little bit later in this presentation about why that might be. So then for the order Coleoptera, the beetles, remember I collected 76 of those. 13% came from family Cerambycidae, which are longhorn beetles. Then we have 12% from family Claridae, which are checkered beetles. That's an ornate checkered beetle on Oregon iris that I saw. Then 66% were from family Meliridae, which are the soft-winged flower beetles. And then there were 9% of beetles that were from some other family, like this ladybug here. Flies that I collected. So 63% of those 43 flies I collected were from family Syrpidae. These are hoverflies. They're oftentimes black and yellow like that. They look a little bit like bees, but you can tell it's not a bee because it only has 2 wings and it has those really big eyes. And those are actually really good if you see them in your garden because they're pollinators on sometimes, and they also are good for pest control. And then we also had 28% of the flies I collected were from family Bombiliidae. Those are bee flies. They're these really fuzzy flies. They like to hover above flowers, and they have these really long proboscises that they stick into the flower to drink the nectar. They kind of remind me of if a hummingbird was a fly. They're also very fuzzy. I like them a lot. And then 9% of flies were from other families. So here's this pollination network that I made. I made this in Microsoft PowerPoint, by the way. It was a challenge, but on the left here are the different insect groups. So in yellow are all the different families of order Hymenoptera, the bees and wasps. The green is the different families of beetles. The blue is the different fly families. Then this orangish brown here is from order Hemiptera. Those are true bugs like stink bugs. And I only found a few of those, so I didn't include them in the previous slide. And then I found a single insect from order Odonata, which is damselflies and dragonflies. I figured I might as well put that in there too, but I'm not going to include that in any of my other data analysis. And then on the right, we have the names of all the plant species that I collected bugs on. So the lines going between each group of insects and each plant shows the number of visits of that insect to that plant. So a thicker line means that more insects visited that plant. So you can see Canada thistle, a lot of beetles from family Maliridae visited there, a lot of bees from family Apidae and from family Halictidae also visited Canada thistle. And this also shows that a lot of the pollinator taxa visit a lot of different flowering plants. They're not specific to one family or one species. So these are some of the most abundant flowers that I showed in the previous slide, showing that Canada thistle had 103 visits, mostly from bees, but also a good amount from beetles. Then common St. John's wort had 30 visits. Oxeye daisy had 44 visits from insects, actually pretty evenly distributed between all four orders. That was really the only plant that had a high level from order Hemiptera. And then the largely sandwort didn't get very many visits, and neither did trailing blackberry or European blackberry. But they got more than some other flowers that were less abundant. So let's return to those 9 most abundant plants. So the common St. John's wort, Canada thistle, cleavers, hedge parsley, largely sandwort, oxeye daisy, European blackberry, small flowering Umatilla, and trailing blackberry. Those are here along the x-axis. The y-axis shows that insect visitation rate that I calculated. So if you remember, that's the total number of insects I collected on each species divided by the total number of flowers for that species. So a high visitation rate doesn't necessarily mean that there were a lot of insect visits, because it could mean that there were just weren't very many flowers and there were a lot of insects. There weren't many insects visiting it either, but that's why I chose to only do this for the most abundant plants. So you can see that the plants that had the highest visitation rate for insects overall were Canada thistle and oxeye daisy. Insects really liked to visit those flowers. Now, I'm going to add a second y-axis to this graph. Don't worry, I will explain what it means. This other y-axis on the other side is showing the total number of blooms for each of these plant species. So each of these dots represents the total number of flowers that I counted for each species below. So I counted 1,330 flowers for common St. John's wort, but it only had a visitation rate of 0.02 for all insects. Now, the yellow bar shows the visitation rate just for order Hymenoptera, which are the bees and wasps, which Hymenoptera really liked Canada thistle. And then the green bar shows order Coleoptera, beetles. Those ones like Canada thistle, okay, they also liked oxeye daisy quite a bit. And the blue is for the visitation rate for flies. They didn't have a super high visitation rate for any plants, but they visited a good amount of these. And you'll notice that the visitation rate does not correspond to the number of blooms. The plants are arranged with the most abundant here and the least abundant there, but the visitation rate doesn't start really high and get lower as you go across, right? If you'd expect that pollinators really liked the plants that were most abundant, you'd expect common St. John's wort to have the highest visitation rate, but that isn't what we found. So the objective of my study was to figure out what flowering plant species are good food sources for pollinators in oak savanna. And what I found was that flower visiting insects really liked Canada thistle and oxeye daisy. And my hypothesis was that flower visiting insects or pollinators, I guess, would specifically prefer certain species of native plants. But these two species are actually invasive. So that is not what I expected at all. But pollinators, or no, I'm talking about flower visiting insects. Flower visiting insects did not prefer only invasive plants. You can see that actually 5 out of these top 9 most abundant blooms were invasive, and only those 2, only Candidacyl and Oxeye Daisy, had that high insect visitation rate. And actually, the 3rd highest insect visitation rate was on trailing blackberry, which is our native blackberry. So there doesn't seem to be a super clear preference for native plants or for invasive plants. So I initially thought that maybe insects preferred those 2 plant species because there are so many of them. They're very abundant blooms. But as we saw with this graph, the bloom abundance does not predict how much insects like the plant. Because again, we'd expect them to like St. John's wort most if they like the most abundant plants. So there's something else. I'm not really sure what it is that makes insects like Oxeye Daisy and Canada Thistle so much. It might be the appearance of the blooms that they like. It might be that they produce a lot of nectar or a lot of pollen, or that the nectar and pollen they produce is really high quality. That is beyond the scope of my study, but it would be really interesting to look into later. So for whatever reason, flower visiting insects really liked Canada Thistle and Oxeye Daisy, 2 invasive plant species. So this got me wondering, can invasive plants sometimes be good for pollinators? So there's definitely a high diversity of flower visiting insects that I found, but that doesn't mean flower visiting insects are not always pollinators, because not all insects that visit flowers are going between flowers of the same species and transferring pollen as they do so. So if we're interested in what plants are good food sources for pollinators, we need to know which of the insects that I collected are actually pollinators. So the largest group of flower visiting insects I collected were bees. And these bees are generally known to be very good pollinators. This is because they actively carry pollen intentionally because they're provisioning their young, their nests, with pollen. You can see here in this photo, this sweat bee is carrying pollen on her legs that she will then bring to her nest. So as the bees are going between flowers, they're trying to collect pollen, carry it with them, and it'll fall off often onto the flowers of the same species. The female bees tend to be the most effective because they're provisioning their nests, but male bees are also often very fuzzy and good at transferring pollen between flowers. This graph, we're going to look at insect visitation rate again, but just for order Hymenoptera, which if you recall, 97% of that is bees. And I've outlined the invasive plants along the x-axis in red. So you can see that bees, or order Hymenoptera in general, was able to have a pretty high visitation rate on invasive plants. That shows that they can probably adapt to getting the nectar and pollen they need from invasive plants like this. Now, there was also this high number of bee genera. There are 14 that I found. And that indicates that the bee community is not only a lot of them, but they're pretty diverse. They're doing pretty well. But it's important to note that not all bees are pollinators. So actually this is from genus Nomada. These are cuckoo bees. So they will sneak into the nests of other bees, especially mining bees like this Andrina. They'll sneak into their nests, lay their eggs, and then their larvae will kill the eggs of the bee that originally made the nest and then eat all the food that bee provided for her young. So they don't collect pollen for that reason because they're not provisioning their own nests. And that might be why most of them were not foraging. I found they might have been sitting around looking for nests to sneak into instead of visiting flowers to collect pollen. But also notice that they're not very fuzzy. See, that mining bee has a lot of hairs. The cuckoo bee doesn't have very many hairs, so pollen's not going to stick to her when she is visiting flowers. So that might be another reason these ones might not be super effective at pollinating. But that doesn't necessarily mean that cuckoo bees are bad. They're actually a natural part of any bee community very often. And their presence indicates resilience and diversity in the rest of the bee community. So it's actually a good sign that I found these cuckoo bees here. And it indicates that the bees that do pollinate, the bee community in general, can adapt to having so many native plants in their ecosystem. But I didn't only collect bees. I also collected insects like beetles. So some beetles do pollinate. They're known to pollinate. Beetles from these 3 families, actually, that I found. Longhorn beetles and checker beetles and soft-winged flower beetles. Some of the species of these families are known to be pollinators. And they're actually pretty effective at transferring pollen between flowers of the same species. Not nearly as effective as bees, but they do provide some pollination services to many plants. So if you look at the pollinator visitation rate for beetles, they also visited these invasive plant species, so they can probably adapt to those plants taking over as well. They're not super threatened by the oxeye daisy and the creeping thistle coming in. Then we'll look at flies that I collected. Most of them were from family Circidae. Those are the most common flower visitors that I found, and they also are the most effective pollinators of all flies generally. The family Bombyliidae, they're not as effective at pollinating. That might be because they kind of hover above the flowers as they're drinking the nectar rather than landing on the flower, like many hoverflies do. So if we look at the visitation rate for flies, they also were visiting invasive plants as well as native plants. So they can adapt too. All of these insect species are able to get nectar or pollen from invasive plants. And that means that they're probably going to be okay, even if the native plants are declining. But just because that's good news for the pollinator community, that doesn't necessarily mean that the entire ecosystem is going to be okay. So remember these native flowers that I thought I was going to find in oak savanna, and I thought pollinators were going to really like those. But the only one that I found was trailing blackbird. None of these other ones. I didn't find a single flower of any of these other species. And that is likely because they got pushed out by invasive plants. So this is a picture of one of my research sites in August. It is covered in Canada thistle. And let me tell you, I had to walk through that along my transects to count the number of plumes. It was really not fun. But I did it, and I don't have to do it again. But we don't want our oak savanna to look like that, because that means there's very low diversity of native plants, and that's also probably not good for other animals that rely on the oak savanna. Remember these guys, all these threatened species with small ranges, they're not going to be happy if the oak savanna plant community is just Canada thistle. The camas pocket gopher is not going to be in a savanna where there's no camas for it to eat. So invasive plants can get spread by a lot of different ways. They can get spread by machinery from logging, or even from restoration efforts sometimes, or from trail maintenance. They can get spread by people visiting the forest, or from animals visiting the forest. They can spread from other nearby areas. So removing these invasive species, preventing them from spreading, is important for these animals as well as for the native plant community, even if the pollinators are doing okay. So the forest plan for the McDonald Dunn Research Forest actually includes a section about conserving oak savanna and other habitats of concern. Right now, they're doing this really cool project in Jackson Meadow, which is shown on this map here. And they're allocating pretty much all of their resources for conservation to this project. It's a very intensive project. But that means that they don't have a lot of funding or other resources to restore other areas of the forest, like my research sites. So if they were to try to do some sort of invasive plant control in the remnant oak savannas, it would need to be very low cost. So I'm talking a bit about different ways of controlling invasive plants. Herbicide is the most common. That's the most commonly used throughout the McDonald Research Forest. It can be specific to certain vegetation types, or sometimes usually just very general. But what happens if you kill all these invasive plants that are taking over the site is suddenly there's a bunch of free space where no plants are. If you don't plant native plants there, then the invasive plants are going to grow right back because they're very competitive. They compete for that space very effectively. One type of herbicide is pre-emergent, which will just prevent any seeds from germinating. That is very effective to prevent the invasive plants from coming back while you plant native plant seedlings, because those will be affected by the pre-emergent. But again, oftentimes the herbicide, you need repeated applications, which can be expensive because you have to pay for the herbicide. You have to pay the person to apply the herbicide. And herbicides can also pose a danger to native plants, to wildlife, to visitors to the forest, and also to the people who are applying the herbicide. So it's a useful tool, but it's generally not advisable to rely on it too much. Another method of invasive plant management is mowing. So this is really good. You get a big tractor mower like this. It's good for open areas that have a lot of just taking invasive plants. And fortunately, my research sites are open areas with a ton of invasive plants. And mowing is generally low cost if you already have one of these machines, which I'm guessing the McDonald Forest, the Research Forest probably has one of these because they do a lot of large restoration projects. And actually, because oftentimes native plants and invasive plants will have different timings of producing seeds, you can time mowing right before invasive plants develop seeds after the native plants have already finished producing their seeds. And that will prevent the invasive plants from reseeding and they'll still be in the area. They'll cut over back from what was mown, but they won't be spreading their seeds elsewhere. But some other important things to consider is that you do have to mow every year if you're doing this strategy just to keep the invasive plants from spreading. And also it's very important to clean the machinery between uses because you don't want to be spreading invasive plants from one site to another from that mower. Another method I want to talk about is prescribed fire. This is used to kill invasive plants, but it leaves behind bare ground often, which again, invasive plants can come right back if you don't plant native plants right away. So historically, when a fire went through, native plants would reestablish, but now there's these invasive plants that can come back a lot faster. And actually some invasive plants are adapted to fires like this. So it could potentially make the situation worse if you don't know what you're doing. So it's generally not advisable to do something like a prescribed fire unless you're going to do a pre-emergent application and then plant native plants right away. And that can be a bit higher cost. So it's good, really good for more intensive restoration, but for a low cost solution, just to keep the plants from spreading in the rest of the forest, mowing is probably the best option. So I'd like to address a number of limitations from my research study. First off is I was not able to calculate the insect visitation rate for a lot of different flowering plant species because they had a low bloom abundance. So I was really only able to calculate insect visitation rate for the most abundant blooms. And a larger study that looked at a greater area could probably find more individual flowering plants or blooms of each plant and calculate the insect visitation rate more accurately than I was able to. I also only collected data in a single year, and because of different environmental conditions between years, my data might not be able to apply to past years or future years, but it does provide a nice snapshot of this 1 year. My sites were also close together, if you recall in the map. They're just in this one part of this research forest, so I can't necessarily say that my results apply to Oak Savanna and the Willamette Valley, but it does provide useful recommendations for management of this specific forest. So to conclude, I wanted to find out which plant species, which flowering plants are good food sources for pollinators in Oak Savanna, and I thought there was going to be specific native plants. But what I found was that the invasive plants, oxeye daisy and Canada thistle, were actually the most sought out, most preferred by insect pollinators in Oak Savanna. I also found that whether a plant was native or not did not predict how much insects like to visit it, which was another thing I was not expecting. I did find a diverse pollinator community despite all the invasive plants. So that shows that insect pollinators can adapt to invasive plants coming in, at least the ones that I found in this site. And I would also recommend doing some sort of management like mowing as a low cost solution to preventing the native plants from spreading, the invasive plants from spreading, even if they can't do a super intensive restoration effort, because maintaining some semblance of a native plant community and keeping that ecosystem as healthy as possible is important for other members of the ecosystem, even if the pollinators are doing okay. In the future, I think it would be really interesting to look more into why the insects liked those two species, the Canada thistle and oxeye daisy, so much. It would be cool to look at the quality and quantity of the nectar and pollen they produce to see if that might be a reason that those insects are visiting those flowers. I'd like to thank you all for being here today and listening to my presentation. Here's the QR code. You can scan this to look at my references, and I would be happy to answer any questions at this time.
Great job, Florian. Does anyone in the room or online have questions for Florian?
Yeah, first of all, great job. I'm curious to know whether, so you looked at whether the plants themselves were native or non-native. Did you also look at whether the insects visiting those flowers, whether they were native or non-native?
I did not because I didn't trust myself to identify them to the species level because all the genera of bees that I looked at were native to Oregon, and I think there's native species from all the families that I found. But I was able to look at them on the species level to determine if the species were native or not. I'm assumed that most of the pollinators that I found, especially the bees, were native. But again, that would be another future research. If we look into that, that would be interesting.
Um, uh, what what do you suppose the impact of honeybees would be on the populations of native bees or of the other pollinators that were?
So I didn't look at honeybees because they were one that I was able to say these are not native pollinators, but it's possible that they could compete for pollen and nectar with the native bees. But because there were so many flowers at my sites and the native bee community seemed to be doing pretty well, I don't think that's happening here, but I can't really say for sure because
Did you observe honeybees there at all?
yeah, I did see honeybees, but I didn't collect.
Okay, it was just excluded.
I think if I remember right, you said you collected twice each day?
At each
How did-
site.
Oh, at each site, yes. How did you decide the collection times?
I started at site number 1 at around I drive to the forest trailhead. I have my bike. I get my stuff on my bike. I bike to my sites. Then I start sampling. So that usually meant I was able to get there around 9 or 10 in the morning. And I would start when I got there, I'd record the time and then 20-minute hand that there, move on to the next site. And then after I did all of those, I would take a break to do the flower surveys, and then I would do the second round of hand netting at each of the sites. So it was kind of just how I could fit it into the day. But I did actually, I will say, I did try to time it for when insects would be active, especially bees, because they don't like to fly when it's too cold. So 10 or 10 a.m. was when the sun would be pretty bright, they would be active, and then I usually stop before 4 with the starting to cool down. Yeah.
What would you conduct in your research with hindsight using the same premise? And how do you think this could alter the study?
What would I have done differently?
Yeah.
I think I would have chosen different research sites that were maybe had more restoration going on, so I could actually have found some of those native plants that I had hoped to find and see what the insect visitation rates were like on those. But another thing that would be interesting to do would be to compare sites like the ones I did look at with restored sites to see the effect of restoration on the pollinator.
Any other questions? Let me see. You're getting a lot of great job. Oh, there is a question. So this is from Scott Mitchell. If you had more native blooming plants present in your sites, do you think that preferences or visitation rates of your pollinators would change?
I think that considering that past studies showed those some native species were really preferred by pollinators, I think they could change. I think that if there had been a high abundance of some of those flowers, then there might have been more pollinators visiting those flowers and less visiting the invasive flowers.
Okay, last call for questions. Thank you from Scott for answering his question. So with that, if we can give Florian a round of applause. We're going to move and transition to the private defense.